Morning, everyone. Today I'm going to talk on cell membrane transport. Permeability of substances depend on lipid solubility and not on the molecular size. The water molecules compounds are generally impermeable and require the carrier-mediated transport. One important function of membrane is to withhold the unwanted molecules. Transport mechanism can be classified into two major categories. One is the passive transportation, another one is the active transportation. Passive includes the simple type, diffusion, facilitated diffusion, and ion through ion channels, which are specialized carrier proteins. Second type is active transportation, which is energy exhausting process. It happens through certain pump proteins that can drive the molecules against the concentration gradient using energy. Solutes and gases enter into the cell passively, driven by concentration gradient. It occurs from higher concentration to lower concentration, and the rate of entry is proportional to the solubility of solute in the hydrophobic pore of the membrane. This process does not require energy and a very, very slow process. Passive diffusion process, it happens through carrier mediated method also. So structurally similar solutes can competitively inhibit the solute entry by binding irreversibly or temporarily binding with these carrier mediated proteins. uh, these proteins operate bidirectionally and they don't require energy, but the rate of transport is more than simple diffusion because of the mediacy of these proteins. It depends on concentration gradient. You can see it here. Facilitated diffusion, facilitated transportation through carrier mediated molecule. This is the pink state. Carrier molecule exists in two states. One is the Pong state and another one is the pink state. So Pong state, it is an active side exposed to the outer side of the membrane. Here the solute binds and binding leads to the conformational change. And in the pink state, then it transforms into the pink state, and here the active site faces interior, releases the solute molecule into the cell and reverts the protein back to the Pong state. Example are glucose and amino acid transporters. Aquaporins, they are water channels that serve as selective pores through which the water crosses the plasma membrane of the cells. They form tetramers in the cell membrane and facilitate the transportation of water. They actually control the water content of the cell. Diseases like your nephrogenic diabetes insipidus is due to the impaired function of these channels. Channelopathies are a group of disorder that results from abnormalities in protein forming the iron pores or channel. A few examples of cystic fibrosis are chloride channel, Liddle syndrome, which is due to the defect in sodium channel, and the periodic paralysis, which is due to the defect in potassium channels. This is a structure shown of the water channel. So these are the cell membrane. In between the cell membrane, this water channel is persisting here. Ion channels, they are meant for quick transportation of electrolytes like calcium, potassium, sodium, and chlorides. Uh, they selectively meant for selective ions and span all through the length, thickness of the membrane. Few are in closed state, few are open, like they are ready for transportation, and few are in inactive state. They are actually the transmembrane protein and they perform the selective transportation of one particular ion. Regulation of activity is done by voltage gated, ligand gated, or mechanically gated channel process. Different channels are available for sodium, potassium, calcium, and chlorides. Transportation through channel is very, very quick. So voltage and ligand gated channels, they generally remain closed, but in response to some stimuli, they get open and lead to the rapid influx of ion down the gradient. Hence, the name gated has been suggested. You can see it in this diagram. Due to changes in voltage, so voltage difference is created, which leads to the opening of the lid and allows the entry of constant ions to take their entry. Here, the stimulus is in the form of ligand. Ligands are opening the lids of this ligand gated channel, and here you can see the opening of gates of these channels by winding of the ligand, leading to some conformational change. So examples of voltage gated and ligand gated channels are acetylcholine receptor, calcium channels, and amylogenin. Ionophores. Ionophores is a molecule that reversibly binds the ions. Many ionophores are lipid soluble entities and they transport the ion across the membrane. They are working like shuttles for a specific ion. They transport the antibiotic, and ionophores increase the permeability of membrane to the ion by acting as a channel former. They temporarily or reversibly bind, create the channel for specific substances. The two types of ionophores which are being identified, one is the mobile ion carrier for valinomycin and another for gramicidin. Both are antibiotics. Clinical application of the channels, the sodium channels, the local anesthetics such as procaine and they act on the sodium channel and they act as a blocker based on the gating mechanism. Point mutations in sodium channels lead to myotonia characterized by increased muscle excitability and contractibility. In Liddle's disease, the sodium channels in the renal epithelium are mutated, resulting in excessive sodium resorption, water retention, and elevated blood pressure. Potassium channels mutations in long QT lead to long QT interval syndrome, lead to the inherited cardiac arrhythmia, where the repolarization of ventricle is delayed, resulting in prolonged QT intervals in ECG. So this is the clinical application of the channels. Chloride channel, the role of GABA and glycine as an inhibitory neurotransmitter is attributable to their ability to open the chloride channel at the postsynaptic membranes. Cystic fibrosis, it is due to the certain mutation in CFTR genes, which is a chloride transporter. So cystic fibrosis transmembrane regulator proteins, when there are certain mutations, lead to CFTR, cystic fibrosis disease. Then retina, the excitation of retinal rods by a photon is by closing the cationic specific channel, resulting in hyperpolarization of the rod cell membrane. This light-induced hyperpolarization is the major event in visual excitation. So this was all about the various types of passive transportation via diffusion, via mediacy of certain carrier protein in the form of various channels, aquaporins, ion channels, etc. Now move on to the active transport. As the name suggests, active means it requires the mediacy, it requires the involvement of energy. At expense of energy, this process happens. So it is a unidirectional, requires a specialized integral proteins called the transporter, saturated at high concentration of solute, and it is susceptible to inhibition. If instead of the actual substrate, some inhibitor is binded, a competitive inhibitor, which is structurally similar, if binds to the this protein, so there will be complete arrestment of the activity of this channel, this transportation. So this is the structure. This is a sort of diagram. It has various sites for ATP winding, for solutes at different inside and outside as well. Depends on type of transporter. The cell has low intracellular sodium and potassium, which is maintained by sodium potassium activated ATPase or sodium pump. So this is the sodium pump operating, its animation is given here. Here is interiorly there are two sides. One is for winding of sodium and one is for ATP winding. Outside there is a potassium winding site. Hydrolysis of 1 ATP leads to expulsion of 3 sodium and influx of 2 potassium ions simultaneously. Sodium binds to pump, leading to phosphorylation of by ATP and releases the sodium. The potassium binding removes the phosphate and reverts it back and potassium is released. So ion transport and ATP hydrolysis are tightly coupled processes. Digoxin and cubane inhibits the pump by irreversibly winding to the pumps. Then secondary active transport, it can be of two types, uniport or co-transport. Co-transport can be again of two types, symport and antiport. So in uniport, you can see it only one-sided unidirectional movement of substance for transportation happens here. One is in co-transport, one is symport. Simp means one movement or a transportation of one substance facilitate the transportation of other one in the same direction. If it is happening in the same direction, it is symport. If one is going out and another is in response coming inside, this is antiport. So these active processes may be coupled with energy indirectly, as you have seen. Primary in the primary active transport, I've showed you the example of sodium potassium pump. In that pump, the energy was ATP was directly spliced up, releasing the energy at the site, and that energy was released for utilized for pumping of sodium and potassium. Here, this is the indirect utilization of energy. Energy is generated in some other coupled process, and that is utilized for secondary active transportation. This is the basic difference. Example of examples of uniport are calcium pump, glucose transporters. For symport, there is a sodium dependent glucose transport, which is inhibited by fluorescein. Amino acid transportation also. Then antiport example is sodium pump and chloride bicarbonate exchange in RBCs. Thank you.